When we are talking about the cell junctions, there are numerous questions coming these days. And this question is asking, 20 nanometer of intercellular gap is seen in which cell junction? One of the All India PG pattern question, DNB pattern question. So what do you think could be the answer? 20 nanometer of intercellular gap, where? As you are talking about it, it will be zona adherence. Then what about macular adherence, which is other name of desmosomes? Macular adherence, which is other name of desmosomes, it is +5. +5 means 25 nanometer. So there it become +5, 25 nanometer. Yes. What about this one and this one? If you are talking about the zona occludens or you are talking about the gap junction, then they are having very narrow intercellular gap. Very narrow. Very narrow. So you're telling that may it be zona occludens or may it be the gap junction, they have very narrow intercellular gap. Yes. 20 nanometer is present at the zona adherence. Maximum intercellular gap is present at the desmosomes. So maximum intercellular gap is present. What is that? That is 25 nanometer at the level of desmosomes, which are also called as macular adherence. Anyway, you are telling 20 nanometer, answer will be zona adherence. Keep that in mind. As you are moving further, you will find that this is one diagram talking about the cell junctions. And this is the intercellular space. You mean to say there is one cell here, the other cell is here, and this is some intercellular space? Yes. And bounded by some cell junctions could be desmosome. It could be a desmosome. What is the gap? 25 nanometer. Or it could be the zona adherence, which is also called fascia adherence, 20 nanometer. So it could be a zona adherence, which is also called fascia adherence, 20 nanometer. What are you talking about? The gap, intercellular gap. This is one cell, this is other cell, this is the gap. And what is the gap at the level of gap junction itself? Gap junction is like a tunnel between two cells. It is like a tunnel between two cells. And why do you need a tunnel between two cells? For the passage of molecules, ions. You can pass the molecules and ions through this tunnel. And it is having some hexagonal structure, which is having connexons, you know. So connexons are some hexagonal structure. Yes. Connexons are some hexagonal structure making some tunnel between the cells. This is one cell, this is the other cell. For the passage of molecules, ions, this is gap junction. And what is the intercellular gap? It's very narrow. It's very narrow. So this is what we want to discuss in more details. See, this is the basement membrane. And on the basement membrane, we are looking at the two cells attaching to each other with the help of cell junctions. So you are telling this is a basement membrane on which you have two cells attaching to each other with the help of cell junctions. Also, we will talk about the cells attaching to basement membrane with the help of some cell junction. It is hemidesmosome. If hemidesmosome is attaching cell to the basement membrane, then what is the desmosome doing? Attaching one cell with the other cell. Let us start with the details. You see, in the beginning, there is very narrow intercellular gap. This is the level of zona occludens, which is also called as tight junction. So when you are talking about zona occludens or tight junction, the intercellular gap is very narrow. It is towards the apical surface. And if you are talking about something below that, then it will be zona adherence, which is also called fascia adherence. So below that is zona adherence or fascia adherence, and the intercellular gap is increasing. How much it has become now? Now it has become 20 nanometer. And what is below that is the desmosomes. Now when you say these are the desmosomes, they are also called as macular adherence. So below that you are showing the desmosomes, which are also called as macula adherens. The intercellular gap has further increased. How much is now? Now it has become 25 nanometer, actually plus 5. And what about the next one? It is again narrowed down. And what is this level? That is the level of gap junction. And what is gap junction? Gap junction is just like a tunnel between the two cells. And why do I need a tunnel between two cells? For free exchange of molecules. Like which molecules you are exchanging? Could be glucose molecule, or could be amino acids, or could be some ions also. So remember that gap junction is working like a tunnel between cells for the free exchange of molecules, and that could be some glucose molecule coming from this cell to that cell, or could be amino acids, or could be ions. Ions could be calcium ion, sodium ion. And what is it for? It is for the purpose of impulse conduction. What impulse conduction? Actually, the gap junctions are like electrical synapse. They are like electrical synapse. So this tunnel is like electrical synapse for impulse conduction. Where? Like some muscles. Which muscles? Could be smooth muscles. Could be cardiac muscles. So you mean to say smooth muscles peristalsis? Yes. And cardiac rhythmia? Yes, cardiac rhythm, cardiac rhythmia. The smooth muscles peristalsis and cardiac rhythmia is maintained by some electrical synapses, which are gap junction. What if they are damaged? If they are damaged, then there will be disturbed peristalsis in the smooth muscles, and there will be arrhythmia in the cardiac muscles. Remember, these gap junctions, which are like a tunnel between cells, conduct ions from here to there, are working like electrical synapses. And these electrical synapses are actually very fast operating, faster than the neural conduction. So they are faster than the neural conduction, and they are seen in smooth muscles and cardiac muscles. As I was telling, they are made up of connexons. I want you to see this diagram. Connexons, hexagonal. So there are some connexons, some hexagonal structure. Yeah, actually sending calcium ion from here to there, sending sodium ion from here to there. Cardiac muscle, smooth muscle, looking like an electrical synapse. If you disturb them, there could be disturbed peristalsis, disturbed arrhythmia, can be cardiac arrhythmia there. Anyway, as you are coming down, you will find that there is a half desmosome here, which is called as hemidesmosome. What is the job of hemidesmosome? The job of half desmosome or hemidesmosome is to attach cell to the basement membrane. So hemidesmosome is attaching cell to the basement membrane, whereas desmosomes are attaching cells with each other. Desmosomes attach the cells with each other. Hemidesmosome attach cell to the basement membrane. And now we have to discuss two clinical conditions. But before you discuss them, you have to talk about cell adhesion molecules. Now, when you are talking about the cell adhesion molecules, there is one which is called as integrin. And what about integrin? Integrin is a cell adhesion molecule used by hemidesmosome so that you can attach cell to the basement membrane. What is a cell adhesion molecule for the desmosomes? Desmosomes, what they have is actually calcium modulated adherents, which are also called as cadherins. So you are telling desmosomes, they are using some cell adhesion molecule which are calcium modulated adherents. Yes, cadherins. What happens in pemphigus vulgaris is there is some autoantibodies destroying cadherins of the desmosomes. So desmosomes are destroyed and there will be intercellular separation. If you remember, we have discussed that earlier as well. So telling that pemphigus vulgaris, which is presenting as some skin blisters, is autoantibodies destroying cadherins of the desmosomes? Yes. And there is some intercellular separation. One sign become positive. Which sign? That is Nikolsky's sign. So Nikolsky's sign is positive in which condition? That is pemphigus vulgaris. Nikolsky's sign is positive. How do we do Nikolsky's sign? That will find out, but it is positive in pemphigus vulgaris. If you compare it with the bullous pemphigoid, you will find the bullous pemphigoid is actually autoantibodies destroying the hemidesmosomes. Now, if you say that bullous pemphigoid is autoantibodies destroying hemidesmosome, it is actually separating the epithelium from the basement membrane. So epithelium will separate from the basement membrane. This is actually a subepithelial lesion. Bullous pemphigoid is a subepithelial lesion, whereas pemphigus vulgaris is intraepithelial lesion. Both are autoimmune disorders. Let us compare them further now, because questions are coming on these topics. So as you are talking about Nikolsky's sign, which will be present or positive in the pemphigus vulgaris, you are actually putting a tangential pressure on normal skin, and there is extension of lesion into normal skin. Putting a tangential pressure on normal skin and extension of lesion into the normal skin, Nikolsky's sign positive. Even the normal skin will develop the same lesion as the area of lesion. And why is that happening? Because there is some intercellular separation. Because there was some intercellular separation. Feature of pemphigus vulgaris. And pemphigus vulgaris, as we have learned earlier also, layer number 4 is involved. What is layer number 4? That is stratum spinosum. So stratum spinosum having some desmosomal attachment, they are destroyed, and then there will be some intraepithelial lesion. This is pemphigus vulgaris. What about the subepithelial lesion, where there are some separation of epithelium from the basement membrane? As the epithelium is separating from the basement membrane, it is some autoimmune disorder destroying the hemidesmosomes, that is bullous pemphigoid. If you're doing some immunocytoplasmic staining, that immunofluorescence basically, you will find that layer number 4 involved in pemphigus vulgaris, which is intraepithelial lesion, there is a fishnet appearance. But if you are talking about bullous pemphigoid, which is subepithelial lesion, the green immunofluorescence is restricted to the basement membrane, basal layers. Should be restricted to the basal layer. That green immunofluorescence is restricting itself to the basal layers. A problem of hemidesmosomes, but it is throughout the epithelium, like fishnet appearance, then it is pemphigus vulgaris. Though layer 4 is specifically involved, but it can be a large area of lesion,